So imagine you've got this master jewel thief, right? But this thief doesn't even have a physical body until the exact moment they pick the lock.
Right, like they're structurally just a ghost.
Exactly, a ghost. But the second they actually touch the vault door, they harden into this unbreakable padlock. They completely bypass all the security systems, and they just walk away with the vault.
It's a great analogy.
Yeah, and today we're actually looking at a real-life biological version of that exact roast. So welcome to the Deep Dive, everyone. We are unpacking a pretty groundbreaking 2026 study from Nature Communications.
Yeah, investigating the hepatitis B virus, or HBV.
Right. And the mission today is to really understand how scientists finally managed to catch this invisible thief in the act.
Which was an incredible piece of detective work, honestly. Because what we're specifically looking at is how a tiny, basically shape-shifting viral protein called HBx completely hijacks human liver cells.
And that hijack is really the fundamental first step, isn't it? Like, it leads to chronic hepatitis B infection and ultimately hepatocellular carcinoma.
Yeah, liver cancer.
Which is terrifying.
It really is. And to understand how the virus gets away with this, we have to look at the structural mechanics of a specific cancer-linked version of this protein, which is known as the HBx1-120 isoform.
Okay, let's unpack this for the listener. Because, I mean, the stakes here are incredibly high. When HBV infects a liver cell, it does something very insidious.
Right, it drops its payload.
Exactly. It deposits this persistent circular piece of viral DNA called cccDNA straight into the nucleus of our cells. It basically builds a little rogue chromosome and just hides it among our own DNA.
Yeah, which is a massive problem for the cell. Naturally, your immune system and your cellular defenses are actively trying to hunt down this rogue DNA and silence it.
Which is exactly what should happen, right? We have defense systems for this.
We do. The human cell has these really robust epigenetic defense systems that are meant to detect foreign, unsilenced DNA and shut it down immediately.
But the virus brings along a bodyguard.
Right, the HBx protein. And when HBx enters the scene, it somehow stops our cellular defenses cold. It allows the viral cccDNA to just remain active and replicate.
And what's wild is that for decades, scientists knew HBx was the culprit, but it remained sort of the dark matter of the virus.
Yeah, it was incredibly frustrating for virologists.
Right, because we know what the outer shell of the hepatitis B virus looks like, but this specific regulatory protein has totally evaded our understanding because it simply lacks a fixed shape.
Right, it's what virologists call an intrinsically disordered protein, or an IDP.
Which is, you know, a very polite scientific way of saying it's structurally chaotic.
Oh, absolutely.
I was reading this study, and the best analogy I could think of for an IDP is a wet noodle.
A wet noodle is actually pretty accurate.
Right. It's just a floppy, shapeless strand floating around in the cell. And it only takes on a specific rigid shape when it physically wraps around a target protein.
Exactly. But that raises a massive, massive logical problem for researchers.
Right, because if we want to design a drug to stop it, we need to know its shape. But if it doesn't have a shape until it hits something?
Right. Then traditional methods for looking at proteins like X-ray crystallography or, cryo-electron microscopy, they completely fail here.
Because they need the protein to sit still.
Exactly. Crystallography requires you to literally freeze the protein into a rigid crystal lattice. So if you try to take a picture of a disordered wet noodle being thrown across a room, you just get a blur.
You cannot crystallize chaos.
No, you really can't.
So how did the researchers in this Nature Communications paper actually bypass that? I mean, how do you map the structure of a ghost?
What's fascinating here is they used a really brilliant combination of cutting-edge experimental tools and computational tools.
Okay.
First, they managed to stabilize this specific cancer-linked isoform, the HBx1-120, without fundamentally altering its sequence.
Right.
Then they relied on a highly sensitive technique called NMR spectroscopy, nuclear magnetic resonance.
Okay, I know NMR.
Yeah, and here's the crucial part. They fed that experimental NMR data directly into AlphaFold 3, the AI structure predictor, to build a cohesive model.
Right, let me stop you there because that combination is fascinating. I understand NMR. You're basically putting the protein in a giant magnet, which lets you track the magnetic properties of individual atoms.
Exactly.
But AlphaFold 3 is an AI. It predicts protein shapes based on their amino acid sequence.
Right.
So if this protein is a shapeless wet noodle by definition, how does an AI predict its structure? Is the NMR data basically like fact-checking the AI's homework?
That is a phenomenal way to put it, actually. Yes. AlphaFold 3 is incredibly powerful, but with an intrinsically disordered protein, it might predict dozens of potential transient shapes.
Because it could technically fold into a lot of things.
Exactly. The AI gives you the realm of possibility. The NMR data provides the physical reality.
Oh, I see.
Yeah, so in NMR, scientists track what we call chemical shifts in the protein's backbone and specific methyl groups.
Which are what exactly?
They're these tiny clusters of carbon and hydrogen atoms on specific amino acids, like leucine, isoleucine, and valine.
So when the floppy HBx noodle finally bumps into a host protein and folds into that rigid shape, the magnetic environment around those specific amino acids suddenly changes.
Right. Exactly.
And the NMR scan picks up that change like thermal goggles.
Yep.
The amino acids literally light up when they make contact, and the researchers can tell AlphaFold, hey, focus on the models where these specific spots are touching.
Precisely. By tracking those chemical shifts, they could literally watch this disordered protein fold in real time as it engaged its targets.
Wow.
And then they just use the AI to generate a highly accurate 3D structural map of the interaction.
Okay, so just to do a quick checkpoint here for you guys listening, HBV hides as the cccDNA in your liver. The HBx1/120 protein protects it, but it has no fixed shape. So scientists used NMR as thermal goggles to watch it fold in real time.
That's a perfect summary.
Okay, so we've caught the wet noodle in the act of folding. The next logical question is, what is it actually grabbing onto?
Right.
Because this protein is busy. HBx is known to interact with over 200 different host proteins in the human body.
It's incredibly promiscuous, structurally speaking.
Yeah, but this study focuses heavily on two main suspects, right? Bcl-xL and Spindlin1.
Yes. Let's address Bcl-xL first, because it's a really critical part of the investigative process, even though it ultimately serves as a bit of a cautionary tale.
Okay.
So Bcl-xL is an apoptosis regulator. Its normal job in your body is to help control whether a cell survives or undergoes programmed cell death.
Now, the NMR data did show HBx interacting with Bcl-xL, right?
It did, yeah.
It forms a connection using this small coiled section called a BH3-like helix.
Right.
But from what I understand from the paper, this particular isoform of the virus, the HBx1/120, has a truncated version of that helix. Like, it's chopped off.
Exactly. So the connection is what the study calls monovalent.
Just a single point of contact.
Right. Which means it's an incredibly weak, very transient interaction.
Well, then I have to push back on this a little bit. If the connection is just this weak, passing handshake, how could that possibly be the main driver for a chronic, lifelong cancer?
Right.
That feels like a decoy.
Your deduction is spot on. And it's exactly the conclusion the researchers reached, too.
Really?
Yeah. For years, the field suspected that HBx's interaction with apoptosis regulators like Bcl-xL was a primary mechanism for disease.
Oh, wow. So they were chasing the wrong thing.
Basically, this structural data proves that, at least for this cancer-linked isoform, the bond is simply too weak to be the main driver of the massive cellular reprogramming we see in hepatocellular carcinoma.
So Bcl-xL is essentially a red herring.
It's a red herring.
Which brings us to the real target. The actual victim of our jewel thief.
Yeah.
Enter Spindlin1.
Yeah, Spindlin1 is where the story gets deeply, deeply sinister.
Here's where it gets really interesting.
Yeah. So Spindlin1 is a human protein that acts as an epigenetic reader. Structurally, it has 3 distinct barrel-shaped sections called tutor domains.
I love the biological mechanics here. So if you, the listener, are trying to visualize Spindlin1, picture a highly specialized cellular librarian.
I like that.
Right. Our DNA is a massive library of instruction manuals. But you can't just have every single manual open at once. Like a liver cell doesn't need the instructions for building an eyeball.
It would be absolute chaos.
Exactly. So the library uses chemical barcodes on the bookshelves to tell the cell what to read. And in biology, these barcodes are methylated histone tails.
Right, and to get slightly technical for a moment, let's decode what those barcodes actually are.
Let's do it.
The text focuses on marks like H3K4ME3 and H3K9E3.
Okay, alphabet soup. Break that down.
Right, so H3 refers to histone 3, which is literally the microscopic spool that your DNA wraps around to stay organized.
The spool. Got it.
The K stands for the amino acid lysine. The number is its exact position on the spool's tail. And the Me3 part means it has 3 methyl groups attached to it.
Okay, so depending on where those methyl groups are placed, it completely changes the meaning of the barcode.
Exactly.
So H3K4ME3 is the barcode that tells our librarian, Spindlin1, hey, this section is open, pull these books off the shelf.
Yes, it triggers active transcription.
But H3K93 is the exact opposite barcode.
Right.
That barcode means lock this DNA in the archives, do not read it. It silences the DNA.
Perfect. And the librarian, Spindlin1, uses its 3 tutor domains, like barcode scanners, to read those marks.
Like actual grocery store scanners.
Basically. The tutor domains are essentially these microscopic hydrophobic cages that perfectly fit over those methyl marks. So Spindlin1 scans the histone tails and dictates the cell's behavior.
Okay.
But when our viral protein, HBx, enters the scene, it doesn't just attack the librarian. It performs an absolute masterclass in molecular mimicry.
It tricks the scanner.
It does. The NMR and AlphaFold data revealed that the front end, the N-terminal region of HBx, folds into a specific hairpin shape. And that shape slots perfectly into Spindlin1's 3rd tutor domain.
Oh, wow.
Yeah, and in doing so, it flawlessly mimics a normal human regulatory protein called Skinia DOC.
So it's literally wearing a fake ID.
Exactly.
It walks right up to the cellular librarian, flashes this counterfeit Spindlin C badge, and just wraps itself around Spindlin1.
Yes. And unlike that weak passing handshake we saw with the Bcl-xL decoy.
Right, the red herring.
Right. This time it grabs Spindlin1 bivalently, meaning it secures itself in 2 different places at once.
So it's a much stronger grip.
It is a much stronger grip. But here is the most fascinating twist in the paper.
Okay.
Even with a bivalent grip, and even wearing a fake ID, the researchers found that something was still missing.
Wait, really?
Yeah. In their test tube simulations, just mimicking Spindlin DOC wasn't enough to create the ultra-tight high-affinity bond they suspected was actually happening in a real, living human liver cell.
So there was a missing puzzle piece. Mimicry alone couldn't explain how the virus permanently hijacks the librarian.
Exactly. Which brings us to the paper's biggest, most groundbreaking structural discovery.
The intermolecular zinc finger.
Yes. So let's shift our visual here from the librarian's barcode scanner to a glowing metallic padlock that's connecting 2 distinct chains.
A molecular padlock. That is a terrifying visual. Explain how this padlock actually snaps shut.
It is a marvel of evolutionary engineering, really.
Yeah.
So to truly secure itself, the viral HBx protein brings 2 specific cysteine amino acids to the interaction.
Okay.
Cis61 and Cis69. Meanwhile, the human Spindlin1 protein inadvertently provides a cysteine of its own, Cis96.
Okay, so 3 cysteines.
And Spindlin1 also provides a histidine, His252.
Got it.
When these 2 proteins collide, these 4 amino acids form a microscopic, perfectly shaped cage.
Well, let me guess. Cysteines have sulfur atoms, right? And sulfur loves to bond with certain metals. What exactly are they trapping in this little cage?
They are trapping a single zinc ion, Zn2 plus.
Boom! The padlock clicks shut.
The moment that zinc ion is captured from the surrounding cellular environment, it acts as a structural pin.
Wow.
Yeah. It physically stitches the viral protein and the human protein together. Zinc has a very specific charge and geometry that holds those 4 amino acids like a tent pole.
Right.
And when that zinc pin drops into place, the floppy, disordered wet noodle of HBx is instantly transformed into a rigid, impenetrable mask that is permanently locked over Spindlin1.
I am looking at this mechanism, and it just seems so diabolically specialized.
It really is.
I mean, you have 2 completely different proteins from 2 completely different species donating highly specific amino acids just to build a shared padlock around a random piece of floating metal.
Yeah.
Is this kind of virus-host teamwork common in virology?
It is exceptionally rare. To confirm just how rare it is,
Yeah.
the researchers actually searched the entire protein data bank.
Which is the global repository for every known protein structure ever mapped.
Right. Out of thousands upon thousands of structures, they found only one other virus that coordinates a shared zinc finger with a host protein in this way.
Oh, wow. What was the other virus?
HIV-1.
Seriously?
Yeah. Specifically, the HIV TAT protein binding to a human protein called cyclin P1.
That is wild.
Coordinating an intermolecular zinc finger with your host is an elite evolutionary strategy. What we're seeing is that HBV is essentially weaponized its sequence.
It's packing a massive concentration of cysteine residues into HBx to create these specialized sticky patches.
Exactly. It is actively harvesting zinc from the human cell to build the very chains it uses to enslave it.
Okay, let's do another quick recap checkpoint for you guys. So HBx ignores weak targets. It goes straight for the cellular librarian, Spindlin1.
Yep.
It blocks it by faking an ID, the SPND mimicry, and then it locks itself in place using a shared ultra-rare zinc pin.
Spot on.
The structural biology is beautiful, but so what does this all mean for the liver cell?
Right.
And for you, the listener? Like, why does locking up a cellular librarian actually lead to a lethal cancer?
Well, if we connect this to the bigger picture, we have to look back at that hidden viral cccDNA we talked about at the very beginning.
The rogue chromosome.
Yes, because HBx is physically masking Spindlin1 with this zinc padlock, Spindlin1 can no longer read those histone barcodes.
Right. Its tutor domain scanners are physically obstructed. It's blinded.
Exactly. Now, in the scientific community, there are currently two competing models explaining how this specific blinding drives the disease pathology.
And these are the Ducreux model and the Liu model.
Right. And they both lead to the exact same devastating result, a thriving virus and a cancerous liver, but through entirely different mechanical pathways.
Okay, let's break down both, starting with the Ducreux model, because when I read this one, it instantly felt like a classic hostage scenario.
A hostage scenario captures the mechanics perfectly, actually.
Oh, good.
Yeah, in the Ducreux model, Spindlin1 is considered a cellular restriction factor, so it's one of the good guys.
Okay.
Normally, our cell's defense mechanisms try to put repressive marks, those H3K9 and E33 barcodes, directly onto the viral cccDNA.
The lock it in the archive barcode.
Exactly.
Right.
This is a distress beacon. It signals other defender proteins, like HB1, to come in, compact the viral DNA, and silence it.
So the normal process is read the barcode, shut down the virus.
Right.
But in the Ducreux model, HBx takes Spindlin1 hostage. By throwing that zinc padlock on it and taking it totally out of commission, the cell loses a critical part of its silencing machinery.
Precisely. The cccDNA never gets properly compacted, it stays active, and the virus thrives unbothered.
Okay, so that is the straightforward hostage scenario.
Yes. But the Liu model is far more insidious.
Okay.
If Ducreux is the hostage scenario, the Liu model represents the accomplice scenario.
Okay, this is the part that genuinely confuses me. I completely understand tying up the librarians so they can't lock the virus away. That makes sense.
Right.
But how on earth does the accomplice model work? How does a virus basically brainwash the librarian to actively help it?
It has to do with how the padlock changes the librarian's structural affinities.
Okay.
The Liu model suggests that when HBx mimics spina and locks on with that zinc finger, it doesn't just blind Spindlin1, it supercharges it.
Supercharges it.
Yeah, the binding actually increases Spindlin1's affinity for bivalent marks. It forces Spindlin1 to abandon its normal duties and seek out DNA that is in a poised state.
Poised state, meaning?
Meaning DNA that is caught halfway between being active and being silenced.
And let me guess, the viral cccDNA happens to be in their exact poised state.
Exactly. So instead of just taking the librarian out of the game, HBx redirects Spindlin1.
It rides Spindlin1 straight to the viral cccDNA.
Yes. And once there, the supercharged complex actively kicks off those silencing HB1 proteins.
Oh, wow.
Yeah, it forcefully rips away the cell's epigenetic defenses, which guarantees the viral DNA remains fully active.
That is wild. So whether Spindlin1 is a neutralized hostage or this active accomplice, the immediate result is exactly the same. The virus's DNA stays active and keeps churning out more virus.
Right.
But we still have a gap here. A replicating virus causes liver inflammation, sure, but how do we leap all the way to hepatocellular carcinoma?
Right, the cancer link.
Yeah. How does this molecular padlock actually trigger cancer?
This is where we see the massive collateral damage of scrambling epigenetic readers. Spindlin1 isn't just responsible for reading the barcodes on the invading viral DNA.
Right.
Its primary job is reading the barcodes on our human DNA.
Ah.
And when you have a chronic hepatitis B infection, the virus is mass-producing thousands of these HBx padlocks, just flooding the nucleus.
So it's throwing the librarian's books off the shelves everywhere, not just in the viral section.
Exactly. It disrupts the entire epigenetic landscape of the liver cell. The cell essentially starts suffering from identity theft.
Wow.
Yeah. When you blind or hijack epigenetic readers like Spindlin1 on a massive scale, it accidentally activates silenced host oncogenes.
And oncogenes are the specific genes that when they get turned on inappropriately, they drive cell division completely out of control.
Right.
They're the engine of cancer.
Yes. Normally, a mature liver cell had its oncogenes tightly locked away in the archives with those H3K9E3 barcodes.
Right, the cell knows its job. It's a liver cell.
Exactly. But when HBx scrambles the system, those barcodes are ignored. The cell forgets it's supposed to be a stable liver cell and starts expressing genes meant for rapid embryonic growth.
Oh, that is terrifying.
It pushes the tissue directly and aggressively toward hepatocellular carcinoma.
That is deeply unsettling. But, I mean, it's also just a testament to the sheer minimalist genius of this virus.
It really is.
I mean, think about what we've mapped out today. We are talking about a tiny, intrinsically disordered wet noodle of a protein.
Yeah.
But it compresses multiple devastating attack strategies into one short sequence. It floats around harmlessly, it ignores decoys, until it finds the exact right target.
Yep.
It fakes an ID, steals a zinc atom from the environment, builds a structural padlock from scratch, and brings the entire cellular library to its knees.
Rewriting the cell's destiny in the process.
It is crazy.
It is without question an evolutionary masterpiece. But, understanding the exact mechanics of a masterpiece is often the very thing that allows us to dismantle it.
True.
And this raises an important question, one that I think points toward the future of medical science.
Okay.
I want you, the listener, to consider this as a final thought experiment. We've established today that this incredibly tight cancer-causing bond between the viral HBx and the human Spindlin1 relies almost entirely on that single shared zinc atom acting as a structural pin.
Right, the molecular padlock holding it all together.
Precisely. So what if we could design a drug that acts as a targeted bolt cutter?
Oh, that is a fascinating angle.
Right. If a future therapy could be engineered to specifically chelate, which is just a chemical term for binding and removing that exact zinc ion from that exact microscopic pocket.
What would be the structural consequence?
Exactly. The padlock snap.
The padlock breaks.
And if that happens, does the terrifying rigid HBx mask instantly revert back to a harmless, floppy wet noodle? And if we can force the virus to drop its shield, would that finally allow the human body's natural epigenetic defenses to step back in, read the barcodes properly, and silence the hepatitis B virus for good?
That is a phenomenal thought to leave on. We spend so much time thinking about, carpet bombing cancer cells with broad spectrum chemotherapies.
Right.
But targeting the actual structural vulnerabilities, like designing a microscopic bolt cutter to break a specific zinc padlock, that truly feels like the future of virology and oncology.
It really does.
It takes the murky, blurry waters of viral diagnostics and gives us a new kind of high-resolution map. It shows us exactly where the break is, and more importantly, it shows us exactly how we might finally fix it.
Well said.
Thank you so much for joining us on this deep dive into the hidden architecture of hepatitis B. Keep questioning, keep exploring the microscopic world, and we'll see you next time.